हेलो दोस्तों, बायोमोलिक्यूल्स में मैं डॉक्टर सुनील पाटिल आपका फिर से स्वागत करता हूं। आज नए लेक्चर के साथ हम नया वीडियो आपके लिए लेकर आए हैं। बायोमोलिक्यूल्स बायोमोलिक्यूल्स के 4 या 5 पार्ट्स हैं। फर्स्ट पार्ट में हम कार्बोहाइड्रेट के बारे में डिटेल में देखने वाले हैं। अगर आपको ये वीडियो अच्छा लगता है तो इसे सब्सक्राइब जरूर कीजिए, लाइक कीजिए या शेयर कीजिए। सब्सक्राइब करने से आपको नए आने वाले वीडियो के बारे में पता चलेगा। बायोमोलिक्यूल का फर्स्ट पार्ट में हम सबसे पहले देखने वाले हैं व्हाट इज बायोमोलिक्यूल। लिविंग टिश्यू कंसिस्ट ऑल द एलिमेंट्स प्रेजेंट इन द सैंपल ऑफ अर्थ क्रस्ट। मीन्स जो सैंपल अर्थ क्रस्ट में होते हैं वही लिविंग टिश्यू में होते हैं, बट रिलेटिव अबंडेंस ऑफ कार्बन एंड हाइड्रोजन विथ रिस्पेक्ट टू अदर एलिमेंट्स इज हायर इन लिविंग ऑर्गेनिज्म देन इन द अर्थ क्रस्ट। कार्बन और हाइड्रोजन का अबंडेंस ये अर्थ क्रस्ट से बहुत ज़्यादा होता है। फॉर द केमिकल एनालिसिस any living tissue grind it in trichloroacetic acid, then slurry is strained through the cheesecloth or muslin cloth. We will obtain the filtrate. The filtrate is called the acid-soluble pool, and the second, which is retentate, means inside the cheesecloth, it is the acid-insoluble fraction, means acid-soluble pool and acid-insoluble fraction. These are two parts are formed of the biomolecules. Two types of chemical compounds found accordingly in the living organism. First type, which found in the acid-soluble pool, they have the molecular weight less than 1000 Dalton. Mostly they are approximately the molecular weight it is found is the 80 to 800 Dalton. They are usually referred as the micromolecules or microbiomolecules. All the carbon compounds that we get from the living tissue, they are called the biomolecules. Second thing is that those which are found in the acid insoluble fraction, they are called the macromolecules or biomacromolecules. Then we are moving to the chemically all living organisms have basic 3 types of macromolecule, or they are called as the polymers of simple subunits. That subunits are monomers, polymers of the monomers. These 3 are, first one is the polysaccharide, or they are commonly called the carbohydrates, means the polymer of monosaccharide. Monomers are monosaccharide. Second one is the polypeptide, commonly called as the proteins. They are polymers of amino acids. And third one is the polypeptide, or commonly called as the nucleic acids. They are polymers of nucleotides. Lipid, they are water insoluble structures. They are small molecular weight compound as compared to the macromolecules. So lipids are micromolecules. Then we are going to see the outline of the biomolecules. Biomolecules can be divided into organic molecules and inorganic molecules. Organic molecules again can be differentiated into the macromolecule and micromolecule. The macromolecules are, we have already seen the polysaccharides, polypeptide and polynucleotide. Micromolecules, lipids and secondary metabolites. Lipid again can be further divided into fatty acids and glycerol. We know fatty acids are saturated and unsaturated. It depends on the hydrocarbon chain, which is fully saturated or with double bond unsaturated. Then secondary metabolite, they are also micromolecules. They consist the alkaloid, phenolics, terpenoids, essential oil, toxins, lectins, etc. Then we are moving to the inorganic biomolecules. The inorganic biomolecules can be divided into these 3 parts: the prime elements, the macro elements and the trace elements. The prime elements we know very well, that is carbon, hydrogen and oxygen. The macro elements, nitrogen, phosphorus, potassium, calcium, magnesium and sulfur. While the trace elements are boron, then the chlorine, zinc, copper, iron, then sodium, molybdenum, nickel, silicon and cobalt. Then about the carbohydrates in detail. The term carbohydrate derived from the French word hydrate de carbone, that means hydrates of carbon. Carbohydrate means hydrates of carbon. They contain the element carbon, hydrogen and oxygen with the empirical formula CH2O. bracket raised to n, means repeating units of CH2O, means bracket raised to n. Many units CH2O के होते हैं इसे इसलिए bracket raised to n है. Here the you can see H2O, means the hydrogen and oxygen ratio is 2:1. Same ratio it is present in the water. The carbohydrate can be broken down or oxidized to release energy. First of all, how the carbohydrates are formed? Mostly they are formed in the process of photosynthesis, that is carbon dioxide plus water in presence of chlorophyll of plant, and with the help of sunlight they form the glucose and releases the oxygen. Means one molecule of glucose when formed, 6 molecules of oxygen are released. This sentence, carbohydrate can be broken down or oxidized to release energy, means glucose, it is glucose plus oxygen, it releases carbon dioxide, water and energy. So energy is released in the form of ATP, while glucose is oxidized. Glucose plus oxygen, it forms the CO2, H2O and energy. We are continuing the same detailed structure of carbohydrate or classification of carbohydrate. Carbohydrate broadly classified into monosaccharide, disaccharide and polysaccharide. The monosaccharides are also called as the simple sugars. This simple sugar depend upon their number of carbon. They are, if 3 carbon, they are called the trioses. The triose example is the glyceraldehyde. If 4 carbon structure is there, they are called the tetrose sugars. Example is erythrose. If 5 carbon sugar, it is called the pentose sugar. Example ribose in the RNAs, deoxyribose in the DNA. Then if 6 carbon structure, they are called the hexose sugars. Example is the glucose, fructose and galactose. And if 7 carbon sugars are there, they are called the heptoses. Example is the pseudoheptulose. Then we move to the disaccharide, means 2 monosaccharide together from the disaccharide. The sucrose, lactose and maltose. Sucrose, it is cane sugar or table sugar, it formed by the combination of glucose plus fructose. Lactose, it is the milk present in the milk or milk sugar, it is glucose plus galactose. While the maltose, it is called the malt sugar also, it is 2 units of glucose. Glucose plus glucose form the maltose. Then we are moving to the polysaccharide. Polymer of monosaccharides called as the polysaccharide. This polysaccharide broadly divided into 2 categories, that is the homopolysaccharide and heteropolysaccharide. When polymer of only one type of monosaccharide, they are called the homopolysaccharide, and different type of monosaccharide in the polysaccharide, it is called the heteropolysaccharide. So example of homopolysaccharide is starch, cellulose and glycogen. Starch in plant, cellulose in the cell wall, the glycogen in the animal storage. All these are made up of glucose molecule only. So they are the homopolysaccharide. And heteropolysaccharide, polymer of different types of monosaccharide. Example is the hyaluronic acid, N-acetyl glucosamine, etc. Then we are moving to the monosaccharide or one by one. Monosaccharide are the simplest carbohydrate. They cannot be broken down to smaller carbohydrate. Monosaccharide containing the aldehyde CHO group are classified as aldoses. Here you can see the figure. This first carbon with the CHO group, it is called the aldehyde group, and the sugar is called the aldoses. Aldoses, example is the glucose, have the aldehyde group at one end. So example is glucose as well as xylose. And those with the keto group, ketone, COO group, classified as ketoses. The ketoses example is the fructose, have a keto group usually at the carbon 2, second carbon, C double bond O, it is the keto group. So sugars monosaccharide are divided into aldoses and ketoses. Due to aldoses and ketoses, they are reduced, they become reducing sugar if they are free. All monosaccharides are reducing sugars due to presence of free aldehyde or free ketone group. These sugars reduce the Benedict reagent since they are capable of transferring the hydrogen or electron to another compound. So the process is called as the reduction and sugar is called as the reducing sugar. We'll see again later in detail. The pentoses and hexoses can cyclize. This is a hexose with the 6 carbon structure. It can cyclize in cyclic form. Glucose form an intramolecular hemiacetal. C1 aldehyde, this is first CH group, that is C1 carbon 1 aldehyde group, and C5 OH group. It joins with the C5 and this CH2 OH remains outside. So CHO group joins to C5. So a cyclic structure is formed. It is called as the pyranose ring. 6-member pyranose ring is formed. It is in honor of pyran. This represents the Haworth projection. It is also called as the Haworth projection. When the hydroxyl group it is below ring, it is called as the alpha D glucose. If it is hydroxyl group it is above ring, it is called as the beta D glucose. So alpha and beta, it is these two forms formed from this structure, linear structure. So we are continuing monosaccharide one by one. First one is the glucose. Glucose is most important source of energy in living cells. In the human blood, its concentration is about 90 mg per 100 ml of blood. Due to small size and solubility, it passes through the cell membrane into the cell. Energy is released when the molecules are metabolized by cellular respiration. We have already seen the glucose plus oxygen. It forms the carbon dioxide, release carbon dioxide, form water and release energy in the cellular respiration inside the cell in the form of ATP by the mitochondria. We have already seen the alpha OH below the ring. It is the alpha glucose, and OH above the ring, it is beta structure. OH above the ring, it is the beta glucose. Then we are moving to the second monosaccharide, that is galactose. It looks very similar to the glucose molecule. They can also exist in alpha and beta both forms. Galactose plus glucose, it forms a disaccharide called as the lactose, that is milk sugar. Glucose and galactose cannot be easily convertible into each other. Galactose cannot play the role of glucose by the respiration, means galactose cannot form the energy. Then third one is the fructose. It is the fruit sugar, that is, it is a keto sugar or ketose sugar or ketohexose. It has a 5-atom ring rather than 6-atom ring. Fructose plus glucose, it form a disaccharide called as the sucrose. So these 5 carbon atoms are there, that is the fructose. This is first, second, third, fourth, fifth and sixth, but 5-sided ring is formed. This 5-atom ring, CH2OH group present on the both sides. It is the difference in the structure. So it is the structure of galactose, only difference is that instead of oxygen here is the carbon. Then we are moving to the next part, that is the disaccharides after monosaccharide. In nature, most sugars are found in the disaccharide form. When two monosaccharides reacts by condensation reaction, means releasing of water or molecule, they form the disaccharide. The bond formed between these two, it is glycosidic bond, which hold two monosaccharide unit together. The process requires energy. The example of disaccharides are sucrose, lactose and maltose. Disaccharides are soluble in water. They are digested in small intestine. These monosaccharide then pass into the blood and through cell membrane into the cell. So disaccharide, with the help of water, they form two monosaccharides. Then we see sucrose as a disaccharide. First one, common table sugar sucrose, we are using daily, has a glycosidic bond or linkage in the anomeric hydroxyl glucose and fructose. Because of the configuration at the anomeric carbon of the glucose is alpha, first carbon of the glucose, that is alpha, and linked with the second carbon of the fructose, so it is called the alpha 1,2 linkage. It is non-reducing sugar since it lacks free aldehyde or free ketose group. Lactose and maltose are reducing sugar. Sucrose is not non-reducing sugar, while lactose and maltose are reducing sugar. The lactose, it is second one, it is called the milk sugar. It is composed of galactose plus glucose with beta 1,4 linkages. The linkage form the anomeric hydroxyl of the galactose. And third one is the maltose. It is also called the malt sugar. A cleavage product of starch that is maltose. Amylose is a disaccharide with alpha 1,4 glycosidic link between the C1 carbon 1 and carbon 4 between the hydroxyl of two glucoses. You can see here this is a glucose molecule, this is another glucose molecule, first carbon of the first glucose molecule, and this is second carbon. This form hydrogen at one side, so it is alpha and 1, and this is fourth carbon of the second glucose, so it is alpha 1,4 linkages. This is the structure of maltose. Maltose means glucose plus glucose. Then we are moving to the polysaccharide. The polymer of long chain monosaccharide are called the polysaccharide. We have already seen the polysaccharide are either homopolysaccharide or heteropolysaccharide. First homopolysaccharide we are going to see is the cellulose. Cellulose found in plants. It is a homopolymer made from only one type of monosaccharide, that is beta glucose molecule. Used to construct rigid cell wall, plant structure tissue, very insoluble, takes long time to digest. Digestion requires enzyme cellulase. Most animal cannot digest cellulase. Nutritionally it is called as the fiber, or it is called as the roughage, which passes through the intestinal tract, important in minimizing the colon cancer. These polymer molecules are straight without branches. Paper made from the plant pulp and cotton fiber is cellulosic. You can see the one hydrogen, it is below the ring, one other hydrogen, it is above the ring. It forms somewhat Z-shaped structure, so it is beta 1,4 linkages. No doubt it is first carbon, it is fourth carbon, but it is both hydrogen are not above the ring, so it is beta instead of alpha structure. So repeating unit of the glucose in beta form only, unbranched structure, it is the cellulose. Then we are going to see the second polysaccharide, that is starches. Starch, plants store glucose as starches in two forms, either amylose or amylopectin. So it is called as the starches. Glucose polymers collectively called starches. Amylose and amylopectin both are homopolymer of glucose. Only there are differences in their structure. We'll see that. The functions as energy reserve, soluble structures, starches available very quickly. Amylose, first we are going to see amylose. It is a glucose polymer of alpha 1,4 glycosidic linkages as represented below. The end of the polysaccharide with an anomeric carbon, means carbon first, that is not involved in the glycosidic bond. This right end is called as the reducing end. You can see the right end, this is first carbon, it is not involved in the glycosidic bond, and it is the aldehyde group is there, it is called as the reducing end, means it can donate the hydrogen. So right hand end of the starch, it is the reducing end, while the left hand end, it is the non-reducing end of the starch, amylose. Then we see the amylopectin. It is a glucose polymer of with mainly alpha 1,4 linkages, but difference is that it having branches form at certain places after 15 to 20 glucose molecule. It is 1 to 6 alpha 1,6 linkages. So branches are there in amylopectin and unbranched is the amylose. It is the difference. Starch form helical secondary structure. In fact, starch can hold 12 molecules in that helical, and it is called the starch 12. So it is blue in color. You can see these both hydrogen at one above the ring, so it is alpha 1,4 linkages, but it form branches at the 6th carbon of the glucose and first carbon of the other glucose molecule. So it is alpha 1,6. Branch point is alpha 1,6, and these are the alpha 1,4 linkages. In this way, it is the structure of amylopectin. Then third one is the glycogen, which is found in the animals. Glycogen is branched biopolymer, homopolymer consisting a linear chain of alpha 1,4 glycosidic bond glucose residue with further chains branching of every 10 glucose with alpha 1,6 linkages. Structure is similar to the amylopectin, but after every 10 glucose molecule there is a branch structure, while in the amylopectin 20 or more after 20 or more glucose molecule, there is alpha-1,6 branches. It is the difference in the amylopectin and glycogen. In the liver or liver cells or hepatocyte, glycogen can compose up to 8% of the fresh weight. Fresh weight of the liver, 8% it consists the glycogen. After meal of the adult person, the 100 to 120 grams are the glycogen found in the liver. So the amount of glycogen stored in the body, especially within the muscle, liver and red blood cell, mostly depends on the physical training, basal metabolic rate and eating habits and intermittent fasting, etc. So this is structure of glycogen, same as the amylopectin. This is alpha-1,6, alpha-1,4 linkages and branching is the alpha-1,6. Then we are moving to the last point, that is the inulin. It is the polymer of fructose only, homopolymer. Then heteropolymer, the more complex polysaccharide, they are heteropolymer, have building blocks with the sugar and amino acid, means amino sugars, chemically modified sugars, for example glucosamine, N-acetylglucosamine, halurine, etc. They are the heteropolysaccharides. Exoskeleton of arthropod, for example, have the complex polysaccharide called the chitin. These complex polysaccharide are mostly. Then we are moving to the biological significance of carbohydrates. Carbohydrate is reserve food as the starch in plant and glycogen in animal. Second thing, glucose is the main substrate for ATP synthesis. It supplies energy for metabolism. Lactose, a disaccharide, is present in milk, provide energy to the babies. Polysaccharides serve as structural component of cell membrane and cell wall. Carbohydrate chain of plasma membrane, glycoprotein and glycolipid usually face the outside of the cell. They have the role in the cell communication, cell signaling and the forming of protective layer of the surface of some cells. So this is about the carbohydrate. Next video we are going to see, that is the protein, in the next time. So have a nice day. See you again. Thank you very much.